Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore autosomal linkage. At the end of this lesson, you will be able to explain the term linkage and interpret and construct genetic diagram, including Punnett square, to explain and predict the result of dihybrid process that involve autosomal linkage. Let's get started. Autosomal linkage. So what is autosomal linkage? So let us start with some backgrounds. In the early 20th century, Thomas Hunt Morgan, or commonly known as Morgan, and his team provided experimental results to support for the chromosomal theory of inheritance by using fruit flies, or commonly known as Drosophila. Morgan discovered that independent assortment does not apply when two gene loci are located close together on the same pair of homologous chromosomes. Why this happened? Imagine that fruit fly has 8 chromosomes in total. So basically, for homologous pair, 2n equal to 8 in its or their diploid number. This includes 3 pair of autosome and 1 pair of sex chromosome, either XX for female or XY for male. However, Drosophila has an estimated 13,000 to 14,000 genes in its genome. So what this value or this figure try to tell us? The gene, this amount of gene, 13,000 to 14,000 of genes are distributed across its 4 chromosome pair, and therefore a large number of genes in Drosophila are autosomal linked. So look at this diagram. You can see that along the chromosome 2, for example, in this diagram, there are many genes located on the chromosome 2. So this kind of gene are known as autosomal linked gene. And in terms of inheritance patterns, it will be different from what we have learned in Mendelian's dihybrid process. So for example, you can see that, okay, the eyes colors, in this case, purple eyes, light eyes, the wing, and so on, they are located on the same chromosome. So what is the definition of autosomal linkage? So when 2 or more genes are located on the same autosome, when we want to analyze these 2 genes, then these 2 genes tend to be inherited together because they are on the same chromosome. So we can see that in the diagram here, for unlinked gene, for example, we learn in the color of the seed in Mendelian's pea plant, as well as a seed structure, they are located on 2 separate or 2 different pair of chromosomes. However, for linked gene, we can see that gene 1 and gene 2, they are located on the same chromosome. So in terms of inheritance pattern, they will always inherit together. So let us look at autosomal linkage and how it affects the Drosophila inheritance pattern. So in Drosophila, traits are controlled by the gene located on the same autosome. Body color, dominant allele B codes for gray body, recessive allele B codes for ebony or dark color body. And wing shape V, dominant allele V code for normal wing, basically larger, longer wing, and recessive V code for vestigial or short wings. By the way, when we want to choose the alphabet, we try to avoid those alphabet when we write it in the capital letter, or when we write it in the lowercase, they are almost the same. Why I choose V in this case? Because in the later stage, I have a diagram which use B and V, and I have no choice to follow. However, in the exam, when you choose the alphabet to write, make sure that those alphabet give us a very distinctive or very different when we write it in the capital letter and lowercase. For example, we can choose A, we can choose what else we can have, for example, E. Okay? Those alphabets are more suitable to be used compared to V, for example, K, for example. Those are not really suitable. It's not encouraged to use those, okay? So when a homozygous dominant fly for both gene loci, homozygous for B and homozygous for V, is crossed with a homozygous recessive for two gene loci as well, homozygous recessive B and homozygous recessive V, or the F1 offspring are heterozygous and display the dominant trait. So due to the presence of the dominant allele B, therefore they are gray body, and with the dominant allele V, all these F1 offspring have normal wings. So if two genes are located, assuming that the gene code for the body color and the wing are located on different chromosomes, so they are said to be unlinked, which is not true in this context, okay? But imagine that these two genes are located on two different chromosomes. They are unlinked. Because they are unlinked, then we have to follow the principle of independent assortments during meiosis, particularly metaphase 1. So the F1 heterozygous, okay, individual can produce 4 types of gamete in equal proportion. So dominant B, dominant V, dominant B, recessive V, recessive B, dominant V, and recessive for both B and V. And these allele combinations, okay, will occur in an equal proportions. So when this individual cross with a double homozygous recessive individual in the test cross, so the resulting F2 offspring will show 1:1:1:1 phenotypic ratio. Let us look at how the Punnett square show us the cross result. So we have F1 fly, which is gray body, normal wing, cross with double homozygous recessive fly, ebony body, vestigia, or short wing. So we can see that the phenotypes for F1 fly, gray body, normal wing, and for double homozygous recessive fly, ebony body, vestigial wing, or abnormal short wing. So you can see that the genotype, because we know that all F1, they are heterozygous, and for the ebony body vestigial wing, it has homozygous recessive for both gene loci. So because due to the independent assortment, so gray body, normal wing, F1 fly will be able to produce 4 different types of gametes genotypes. However, for homozygous recessive or double homozygous recessive fly, they can only produce recessive B, recessive V gamete genotypes. Let us look at the Punnett square when we cross these two gametes together. So we can see that the Punnett square shows us the flies, or the offspring from the cross shows the equal ratio of 1:1:1:1 for gray body, normal wing, to gray body vestigial wing, to ebony body, normal wing, to ebony body vestigial wing, in the ratio of 1:1:1:1. So this explain that, or show that the results show that independent assortments take place during metaphase 1. So that's why we have that 4 different combinations of gamete genotypes, okay, for F1 heterozygous fly. However, if complete autosomal linkage occur, the two genes are located very close together on the same autosome, and they are inherited together. So look at the diagrams here. dominant allele B and dominant allele V, they are located on the same chromosome. Recessive allele B and recessive allele V again located on the same chromosome. So therefore, during segregations of homologous chromosome take place during meiosis 1, we can see that dominant allele B and V always together, and recessive allele B and recessive allele V, again, they are always together. So in this case, the F1 heterozygous individual will produce only parental type gamete. So what's meant by parental type gamete? So basically, you can see that in this case, parental types always have capital letter or dominant allele B, dominant allele V, recessive allele B, recessive allele V, respectively. So in this case, you can see that the gamete produced in this case will be dominant B, dominant V, or recessive B, recessive V. There is no recombinant type. Okay, because recombinant types of gametes, for example, dominant B, recessive V, recessive B, dominant V, those can only be produced during independent assortments. However, independent assortments can't take place when two genes are located on the same autosome, or we say that on the same chromosome. So therefore, the inheritance patterns will be different from the Mendelian dihybrid process. So a cross with a double homozygous recessive individual will produce 1-to-1 phenotypic ratio in the offspring rather than 1-to-1 to 1-to-1, again, because there is no independent assortment during between these two genes, B and gene V, during metaphase 1, again, because they are located on the same chromosome. So let us look at the cross between the F1 fly and the double homozygous recessive fly according to the autosomal linkage context. So again, the F1 fly is gray body, normal wing flies. Double homozygous fly will be ebony body and with vestigial wing or abnormal short wing. Now, when the question, if question tells us that autosomal linkage take place, then we have to use a bracket to show that capital or dominant allele B, dominant allele V are always together, recessive B and recessive B always together. However, if question didn't mention anything about autosomal linkage or link gene, even though we know that these two genes are linked, particularly questions talk about Drosophila, we know that these two genes most likely they are linked, but we still will assume that independent assortment take place. Clear? Okay, this is very, very important. So I repeat here to emphasize that unless question mention autosomal linkage or question mentions about linkage or link gene in this case, then we will answer the question by using this way. Put a bracket to shows that these two genes are linked. For example, in this context, capital B represents the dominant allele B, dominant allele V together. Another bracket to show recessive B together. And on the other hand, if question didn't mention anything about autosomal linkage, we assume that independent assortment between these two genes occur. We will use the usual Mendelian dihybrid process way to answer the question. So because in this context, dominant allele B and V, or dominant V together, recessive B and recessive B always together. So therefore, in terms of gamete genotype, only two types of gamete genotype, which are parental types. Okay, so compared to the independent assortment cases where we have the recombinant type. So how about these double homozygous recessive flies? Because these flies has recessive B and recessive V, so all the gamete genotype will be recessive B, recessive V only. Let us look at the Punnett square, how the cross between these two flies, and we can see that, yeah, we only have these two phenotypes, okay? So either gray body, normal wing, or ebony body, vestigial wing. And both of them, they are the parental types. Still remember our parental types? We started with the gray body, normal wing, cross with ebony body, vestigial wing. So in this context, the F1 flights cross with a double homozygous recessive flight based on the context of link gene. We won't be able to get 1:1 to 1:1 ratio according to the Mendelian dihybrid ratio, but we will get 1:1 ratio of gray body, normal wing to ebony body, vestigial wing. Okay, so the ratio again 1:1 instead of 1:1 to 1:1. However, based on these two predicted results, Morgan's carried out investigation, and Morgan's experiment shows the offspring from his experiment also included some gray body vestigial wing, which is a mixture of crinoids, all right? The parental types, we have gray body, normal wing, but parental type does not have gray body, vestigial wing. Another phenotype, you can see that some of this flight shows ebony body, normal wing. And these phenotypes occur in a small number, okay, compared to the parental type. So these two types of phenotype is known as recombinant type. It's due to the formations of the recombinant type gamete, okay? So these flight were recombinant types having inherited, okay, a recombinant type gametes from the heterozygous F1 parents. Now, what does this mean by recombinant type? So if you look at these diagrams, okay, we can see that in terms of the formations of the gametes, majority of the gametes are parental types, where dominant allele B with dominant allele V together, or recessive B with recessive V. Those occur in the higher number. So that's why parental types offspring, gray body, normal wing, as well as ebony body, vestigial wing, occur in the large number. However, during prophase 1, crossing over may take place, and crossing over may not always take place between these two genes. So therefore, there are small number of the recombinant type gametes where we can see that dominant allele B now together with recessive V and recessive B with dominant V, they occur in a small number. So the number of recombinant gametes is usually less than the number of parental type gametes. This explains why parental type offspring has higher occurrence compared to the recombinant type. So therefore, recombinant type and parental gametes are not produced in 1:1 ratio. So crossing over does not always occur between these two link gene loci during every meiosis, because crossing over can take place at other location rather than the B and the gene V. So based on the cross, we can actually carry out crossing over value. So what is crossing over value? The crossing over value is the percentage of recombinant offspring produced in a genetic cross. So it reflects the proportions of recombinant gametes that were formed through crossing over between two link gene loci during meiosis, particularly prophase 1. So the formula to calculate this crossing over value equal to, so we take the total number of recombinant offspring So in this context, we're going to sum up the number of recombinant offspring, which are gray body, vestigial wing, recombinant, right? And also ebony body, normal wing, over the total number of offspring observed, multiplied with 100%. So come back to these diagrams again, we can see that the gray body, normal wing, with the dark or ebony body, vestigial wing, those are parental types, due to the parental gametes. So expected result initially show 1:1 to 1:1 ratio, but it is not true because these two genes are linked gene. Okay, so gene code for the body color as well as the wing shape are located on the same chromosome. Okay, so therefore, due to the higher occurrence of parental types gametes, therefore the actual result shows that the parental type offspring occur in higher number. So in this context, 965 and 944, and both of them almost 1:1 ratio. However, when you look at these, a small number of recombinant types gametes are formed due to crossing over. So therefore, because of the crossing over, dominant B now pair with recessive V, and recessive B pair with or combine with the dominant V. And because the occurrence of crossing over cannot be determined, it's not 1:1 ratio in this case, okay, it may or may not occur. So therefore, the percentage of recombinant type gamete is much lower compared to the parental type gamete. So therefore, they produce a small number of recombinant type offspring. So in this case, the recombinant type offspring, we can see that, again, they are gray body with vestigial. So it's something like mix and match in this case, right? So another recombinant type offspring, ebony body with normal wing. And again, they occur in a very small number. So how we can calculate the crossing over value? So we get the summations of recombinant type gamete, okay, divided by the total number of offspring observed. So we can see that we have 206 plus 185 of recombinant types offspring over the total 965, 944 plus 206 plus 185, we will be able to get 17%. So this is the crossing over value. So what is the functions of crossing over value? So crossing over value actually reflect the distance between two linked gene loci on a chromosome. So higher crossing over value indicates that these two genes, they are further apart. When they are further apart, there is a greater chance of crossing over occur between them. We can always imagine that you are provided with two long rope or string and ask you to tie a knot at two ends, will be easier for us to tie the knot compared to the short, okay, string that I give to you guys. If try to imagine we have a very short string and when you learn to tie a knot at the two ends, there is difficult. So the idea here, same or similar here, higher crossing over value indicates that these two genes are far away. So on the other hand, lower crossing over value indicates that the genes are closely linked, making it less likely for crossing over to separate them during meiosis. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment sections. See you in the next lesson. Have a great day.